Good afternoon, everybody. My name is Dr. Kate Quigley, and I'm a senior research scientist at Minderoo Foundation. And today I'll be speaking with you about taking a deep dive into Australian corals of the Aquatic Symbiosis Genomics Project. First, I'd just like to give an acknowledgement of country. So I'd like to acknowledge the traditional owners on the land that we meet today virtually and pay my respects to elders past, present, and emerging. So we know that we're currently in a biodiversity crisis, and current rates of extinctions are approximately 1,000 to 10,000 fold higher than natural rates. And when we look across key animal groups like mammals, birds, and fishes, we can see that some of the endangered and vulnerable species are starting to approach some of the big 5 mass extinction events. So that's where we lost 70% of species on Earth. And so it really suggests that we're getting at a crisis. And unfortunately, Australia is at the forefront of a lot of these extinctions. And this really suggests that we are in a critical time to protect different species. So we know that genomic tools have revolutionized how we understand nature, but how have they really done this? So DNA sequencing can help us to characterize biodiversity, inform different conservation efforts, catalog millions of undiscovered species, reveal the impacts of climate change on biodiversity, either through speciation events or extinction events, and it can help us to conserve endangered species through genomic selective breeding programs. And it also allows us to better predict how particular species will respond when managers apply different wildlife decisions. And this is all in the hopes that we can use genomic tools to preserve and even potentially enhance different ecosystem services by protection of particular species or groups of species. So how can we do this? So one of the tools that we have in our genomics toolbox is something called a reference genome. And this is really the species blueprint, or the idealized assembly of all those different A, T, Cs, and Gs of different individuals within a species. And this is often compared to having an encyclopedia of all the knowledge that makes up that species. And another tool that we can use is something called genome annotation. So if we take this kind of book analogy to another step, the words on the page represent different genes, but the meaning of those words essentially represents their function. So genes essentially transcribe and translate into proteins, and those give the different organism the different functions that it can have. And this is often done through something called de novo assembly, where you need to create that genome for the first time where there was no genomic resource available. And so by having something like a reference genome, which is that species blueprint, we're able to ask questions about why certain species are more adaptable, why they're more resilient, how they breed with each other, as well as beginning to understand individual responses to different stressors. So we know, looking across the tree of life, that global biodiversity is not really covered by different reference genomes. So there's a real lack of resources there. And we know that if we're going to develop species conservation plans from a genetic perspective, we're really going to need to supplement with a number of reference genomes that are available. And if you have enough reference genomes, you can even start to compare across different species, like in this super tree of different coral species represented here. So we founded the Coral Symbiosis Genomics Project, and this is with a number of different collaborators across the world, including Japan, Germany, United States, Australia, where we've all sent our coral samples to the Wellcome Sanger Institute in the UK as part of the Tree of Life program. And so we've had some early outputs, including a manuscript outlining why we would like to do this, and that's called Extending the Natural Adaptive Capacity of Coral Holobionts, as well as a manuscript on how to do it. So we provide consensus guidelines and standard operating procedures for scientists on how they can collect their samples in order to contribute to this initiative. So why sclerotinian corals? So we know they diverged about 500 million or 400 million years ago, and we know that they're key habitat builders to support other biodiversity in the tropics. And this can either be through providing actual habitat for other species or trophically by providing food. And the coral species and the reefs that they make up is essentially built on a rich and dynamic symbiosis between the coral animal and dinoflagellates that live inside of the corals. And those are from the genus Symbiodinaceae, which I'll talk about in a second. And what we know is that although there are a few coral and symbiont genomes out there and the number of them is growing, it just still doesn't represent the total diversity with the hundreds of species that are currently recognized across the world. So if we look a little bit more at this idea of symbiosis, again, thinking about those little microscopic Symbiodinaceae that live inside of the coral tissue that you can see here, these little brown dots. And we can look across different groups of Symbiodinaceae. Here, just a condensed version of different important genera, including heat-tolerant genera from heat-tolerant types from Durastinium. And so this really led to what our project goals were, which was to produce freely available genomes from 41 different coral species and 41 of their different symbionts. And by creating these genomes, we're able to ask questions about comparative genomics, reconstruct early animal evolution, start to untangle uncertainty around coral and symbiont taxonomy. And by identifying key coral and symbiont pairings, we're also able to understand key genomic adaptations that even allowed for the diversification of different symbiotic systems. So in order to do this, we went across the Great Barrier Reef. Here were some collection sites in the far northern part of the Great Barrier Reef. And we collected corals and their symbionts that represented different symbiont transition modes, as well as different reproductive strategies. So corals are really interesting animals because they reproduce in a bunch of different ways and they acquire their symbionts in a bunch of different ways. So we can have spawning species like Montipora digitata or Acropora tenuis, which release their egg and sperm bundles into the water column, where they're broken apart at the surface of the water and they fertilize up there and develop into larvae. Alternatively, we have brooding species that can take in sperm from the surrounding water, and they have fertilization internally, and they brood their little coral planula larvae, which are then released into the environment. And these larvae can either take up symbionts either through horizontal transmission, where they acquire symbionts from the free-living pool in the environment. And this can occur either at the larval or juvenile stage. And then we can have vertical transmission, where the symbionts are actually passed from the mother colony into the eggs in spawners, or alternatively, in brooders, the symbionts can be passed into the planula larvae. So we selected a bunch of different species from across those different types of reproductive and symbiont modes. And in 2019 and 2020, we had two different expeditions. And we were able to go to many different reefs across the northern GBR in collaboration with the Indigenous Partnerships team. We were able to work with traditional owners in order to go to their reefs. And we also worked with the Museum of Tropical Queensland, and in particular, Dr. Tom Bridge, in order to try to collect species that were as close as possible to those museum recognized species or topotypes in order to try to get the taxonomy as correct as possible. And then we also 3D scanned those corals and deposited them into the museum for further work and collection. And so with that, I'd just like to say that we're looking for students on these projects, and please get in touch if you have any questions. Thank you. Bye.